Plant tropism describes how plants respond to stimuli. Like animals, plants produce hormones, or signaling molecules, that are used throughout their bodies by binding to receptors to trigger responses in target cells. However, signaling molecules are slightly different in plants and animals, which is why they're sometimes called growth regulators in plants. Unlike animal hormones, some plant hormones only function locally. Some, like glucose, occur in very large concentrations, and plants do not have blood to facilitate transport. One of the most important growth regulators in plants is called auxin, a class of plant hormones. Its main function is to promote the elongation of plant cells in developing shoots. Auxin is only produced at the shoot tips of a plant and always travels down from cell to cell. It stimulates growth only at specific concentrations by following a reception, transduction, and response signaling pathway similar to ours. According to the acid growth hypothesis, auxin increases the activity of proton pumps embedded in the cell membrane. This causes hydrogen ions to be pumped out of the cell, therefore increasing the magnitude of its membrane potential due to the loss of positive charge inside the cell, resulting in increased ion uptake into the plant cell. Acidifying the cell wall activates expansins, proteins that separate cell wall microfibrils, allowing cell wall loosening enzymes to slide the microfibrils. This results in the cell wall being more flexible. Due to the lower water potential inside the cell because of ion uptake, water osmotically flows into the cell, leading to increased turgor or rigidity of the cell. With the cellulose loosened, plant cells can elongate. Here are some other important plant hormones.
Three main types of tropism, and the first one I'm going to talk about is gravitropism. So as we all should know, plants are very dependent on light as an energy source, but before they're able to grow out of the soil and be exposed to the sunlight, plants depend on gravitropism in order to grow. Gravitropism is a plant's response to gravity on the stems and roots. Gravitropism also helps horizontally placed plants to grow vertically. Gravity is essentially a cue for a plant to grow up when underground and for its roots to grow down. Roots exhibit positive gravitropism, which is when growth is aimed away from the stimuli, while shoots exhibit negative gravitropism, which is when growth is aimed towards the stimuli. So here is the basic structure of the root tip of a plant. So located in the root cap are the statoliths. Statoliths are a type of amyloplast, which are special starch-containing cell organelles. They sink to the bottom on cells because of the gravity. This may also be associated with the detection of a gravitational field. The hormone auxin is also involved in this response. Its action depends on the relative concentration and the targeted organ, whether it be stem or root. So the role of auxin depends on the following. If a stem is horizontal, then auxin produced at the root tip will move down the stem and concentrates on its lower side. Since auxin stimulates cell elongation, growth of the lower is greater than that of the upper side, and the stem bends upward as it grows. If a root is horizontal, then auxin is produced also at the root tip. However, instead of auxin moving away from the root tips and concentrating on the lower side of the root, the concentration of auxin is there to inhibit growth and therefore will behave it in order for roots to bend down. In a normal cell, like the one seen here, the positive tropism to auxin enables the stems to grow up away from the stimulus, while the negative tropism makes the roots grow down towards the stimuli.
Next type of tropism is called thigmotropism, and thigmo means touch in Greek, so thigmotropism is the directional growth in response to touch. For example, vines have the ability to wrap around objects due to thigmotropism. The context stimulates a coiling response caused by differential growth cells on opposite sides of the tendril. The touched cells produce auxin and transport it to untouched cells. Some untouched cells will then elongate faster, which is how the plant can bend around the object. Some plants exhibit rapid leaf movements in response to mechanical stimulation. When this occurs, potassium ions are lost and therefore water leaves the cell by osmosis because the cell would have a higher water potential than its surrounding. This could be a possible evolutionary advantage for the plant because it would not look as leafy or appetizing for its herbivores. Adjacent leaflets can respond in this plant Mimosa pudica on the left due to action potentials. Plant action potentials are much slower than animal nerve impulses, but this mechanism allows the plant to respond to mechanical stimuli much faster than the first process involving auxin. The speed of the response also depends on how violent the stimuli is. For example, if you hit the leaf with your finger, it will respond slower than if you hit it with a hot needle.
The next type of tropism is phototropism, which is the growth of a plant shoot toward or away from a light. The sides of the plant that are not facing the light source elongate, causing the cell to curve toward the light. The idea was created by our old friend Darwin when he was studying a grass that curves toward the light. He noticed that when the tip, called the coleoptile, was removed or covered by an opaque cap, the plant did not curve like it did when the tip was exposed to the light. He also saw that when the base was covered, there was no effect. Next, he tried separating the tip with a permeable gelatin barrier, which had no effect, and with a non-permeable mica barrier, which impaired curvature. Using this information, he postulated that a signal was transmitted from the tip downward, and this caused the plant to curve. Researchers then determined that a chemical, later called auxin, was produced in the coleoptile when exposed to light, and this is what signaled the elongation. The auxin moves through the plant and becomes more concentrated in the darker region, causing the darker side to elongate and the plant to curve. This curvature exposes more of the tip to the light, increasing photosynthesis. The auxin travels from tip to base, and this is referred to as polar transport. The direction is based on the distribution of auxin transport proteins in the cells and does not depend on gravity. The auxin is concentrated at the basal end of the cell, and auxin transporters move the hormone out of the cell and into the apical end of another cell. It moves at approximately 1 centimeter per hour. Auxin is used in controlling the development of the plant. It can pass information, such as showing that one branch is not being productive and that more branches are necessary. Auxin has other uses, including being used in vegetative propagation of plants as well as in herbicide. The reception, transduction, and response of phototropism is the following. Blue light is received by the receptor prototropin, a protein kinase that helps with the curvature of the plant. The response of phototropin is that accumulation of auxin on the shaded side of the stem. The auxin binds to receptors such as TAR1 in the plasma membranes, which causes the phosphorylation of many protein kinases in the transduction pathway. This degrades ARF transcription, repressing proteins, allowing the cell to transcribe enzymes that allow auxin to move to the shaded region of the plant.
Also, this process called etiolation and de-etiolation. And etiolation is a morphological adaptation for growing in darkness, and de-etiolation is greening once the plant encounters light. For example, a potato plant allocates as much energy as possible to elongate its stems, which enables shoots to break ground before nutrient reserves intoned are exhausted. This is an example of how plants' morphology and physiology are tuned to its surrounding by complex interactions between environmental and internal signals. De-etiolation is also known as greening, and it occurs when a shoot reaches light. When this occurs, stem elongation slows, leaves expand, roots elongate, and the shoot produces chlorophyll. The plant starts to look like a typical plant. In reception of deteliation, a phytochrome, a member of a class of photoreceptors, acts as the receptor which responds to light and deteliation. Transduction of deteliation involves the release of second messengers, calcium ions and cyclic GMP, and the signal is amplified exponentially as they activate protein kinases, which usually lead to the phosphorylation of transcription factors. These enzymes transcribed help in function in photosynthesis directly, are involved in supplying chemical precursors necessary for chlorophyll production, or affect the levels of plant hormones that regulate growth. Levels of auxin decrease after activation of phytochrome, which slows stem elongation that accompanies deteliation.
Now we'll look at one of the most beautiful examples of plant tropism working in nature. It involves the rarest flower on earth, the tulip.